Hello, my name is Austin Buskirk, and I'm going to be presenting on the PIP5K isozyme and their impact on HGF-stimulated cell migration. So PIP5K is a critical enzyme that is involved in the generation of PIP2, which is a critical component in HGF-stimulated epithelial cell migration and integrin recycling. The PIP5K gene has various isozymes that include the PIP5KA, PIP5KB, and the PIP5KC. So in order to determine which of these specific isozymes is most responsible for the synthesis of PIP2 during migration, which, as I said earlier, is a component that is critical to the stimulated epithelial cell migration, I performed a series of gene knockdowns of the various isozymes, and I also completed a series of assays in order to determine the impact of each of these individual isozymes on the cells as a whole. So now going on to how exactly I complete these assays and do these knockdowns. So starting with the knockdowns, what I do is I actually use a chemical component called interferon. So what this interferon actually does is it creates gaps and holes within the cellular matrix so that way different siRNA, which are different components that can inactivate certain genes, can actually get through. So by using different siRNA that target these different genetic knockdowns, I can actually knock down the 5Ka, 5Kb, and 5Kc isozymes separately so that way I can see the direct result of which one each does. Another thing that I do with the siRNA is I actually incorporate a non-targeting siRNA so that way the results can be extrapolated and we determine that it's not necessarily the interferon or the siRNA structures that are giving the cells a beneficial response to make them more or less likely to grab onto the fibronectin concentrations. Now, speaking of the fibronectin concentrations, one thing that's really cool with what I do is I actually use varying amounts of fibronectin concentrations, which we will talk about a little bit later during my results stage. So what these actually do is I will take a 10 microgram per milliliter concentration of fibronectin, and I will actually serially dilute it all the way down to 0.1 micrograms. So then I will actually take this and I will put these all in different wells, and then for the next step, I will actually take the cells that I just knocked down, and I will put a certain amount of cells, which is going to be constant throughout. It's typically around 4 times 10 to the 6th power, so about 400,000 or 4 million cells will be put onto the plate. And what I do then is I will go and I will actually fix them with paraformaldehyde. So once the cells are fixed onto the plate, I will then use crystal violet to stain these cells. And this is just to make sure that I can see the cells and then make sure it actually gives them a bright purple color, which is another cool thing that you'll actually see during my results section. So once the cells are actually stained a certain color, we can go ahead and lift the color outside of the cells, after washing it of course, and we can, with deoxychlorate, and what the deoxychlorate does is it pulls it outside of the cells. And once we actually do an optical density reading of this value, it gives us a quantitative value for how much color is in each cell. So in theory, the more cells that are in a well, the more color it will have, and the higher optical density reading. So we can actually directly correlate the optical density reading to the number of cells, and therefore the number of connections the cells can create. So as I was saying earlier, we actually can stain the cells, and then once the cells are stained, typically we go ahead and we lift it with the deoxychlorate. However, in this image here, you can actually see all the cells before we lift it, just so you can kind of see a little bit more of a qualitative representation of the different value or different concentration of cells on the plate. So once the cells have been lifted with the deoxychlorate and the color is in the solution, not in the cells anymore, I actually go ahead and I do an optical density reading of these cells, and I record the optical density and actually compare it to the fibronectin concentrations for each different isozyme knockdown. So this little section here actually includes the data of the most recent trial that I completed. And while I am only including the most recent trial, this is actually due to the fact that the procedure for this sample has actually recently changed, as I have been optimizing this procedure throughout this past semester. So the values that are represented now have been typically consistent within the past trial or two. However, they're not necessarily consistent with the results from earlier in this semester or even last semester for that matter. However, according to the results obtained through this specific trial, I would assume that the PIP5KA or the PIP5KB would more than likely be the isozyme directly responsible for the most adhesion formation. And this is simply because the cells without these two isozymes typically needed a higher concentration of fibronectin to create the cellular adhesions that they needed. So, as I previously touched on, the procedure for this experiment has actually recently changed, and the data isn't necessarily as large of a sample size as I would like to make extrapolations of the results. So, that being said, further research that I would like to do with this experiment would actually be to go ahead and collect a lot more data so that way I can directly analyze it and determine whether or not that this specific trial was just a fluke or if it's consistent with other findings and other results from other trials. Another thing I would like to do with this specific experiment is to narrow down the fibronectin concentrations for each specific trial to see if I can more or less find the optimal concentration of fibronectin for each trial and therefore see the actual peak and make a more quantitative decision on whether or not the specific isozymes bind at better concentrations. So I began my research process here at Penn State by first off reaching out to Dr. Santee, and once I got accepted into the specific lab, Dr. Santee sent me a whole bunch of research papers on our specific topic. So once I read this research paper, I came in the first semester and I learned a little bit about the cells and a little overview of the protocol. So after the first semester, I actually went and did research on my own, and I actually wrote a little bit of a paper as an end of semester debrief. So this end of semester debrief was really, really important in building my foundation for the PIP5K response system and PIP2 recycling, which was really, really cool. So once I am done with Penn State in my undergraduate, I'm hoping to go to medical school or go on to do an MD PhD. So my experiences here will be really, really fruitful and really, really important should I do decide to go on to pursue an MD PhD, because they'll give me a strong background in research and the research processes that I am currently involved in.